Hello viewers, welcome to my channel from Biotechnology tutorial and Research Lab. Today I am presenting or discussing regarding stem cell, genetic or tissue engineering, biotechnology under life science. First of all, I can discuss what is stem cell. Usually in short, I can say stem cell is a cell to develop into a specialized cell type. It can be animal cell, plant cell and human cell. Then what is cell generation? You can see here, in short, the process of reproducing cell in the laboratory. This is called cell regeneration. Stem cell laboratory, nowadays scientists are doing research in a same lab as cell culture laboratory, having animal cell, human cell and plant cell or tissue, because all are correlated to the environment. This belongs to the life science laboratory or stem cell biotechnology, like medical. So these cells are very important, and all cells like animal cells, human cells and plant cells are correlated. As for example, medicinal plant cell, human cell, animal cell, means stem cells can be used in different purposes, like or organ replacement or transplantation. Plant stem cells can be used as biomedicine productions. Animal stem cell can be used for different testing prior to application to the human. Sometimes it is difficult to collect human and animal cells immediately. In that case, plant cell can be used suddenly, and which is available and can be seen whether the new chemicals, you know, innovatives or hormones, new hormones are toxic or non-toxic due to the concentrations after application to the plants, and rapid action can be achieved. Then it can be applied to the animals and human. As for example, in biomedical science laboratory, this animal cells, plant cells and human cells can be used. Some countries it is still different due to the lack of facilities, but in developed countries, scientists are using the cells, means plant cells, animal cells, and human cells, to do cell culture in the same cell culture laboratories. So plant cells, human cells, and animal cells, and microbes, cells or samples. Sometimes it is necessary and correlated. So plant, animal, human, and microbes are correlated and useful to the life science. So this is the background of the stem cells. So now we can see here animal cells and human cells, and also this is called tissue or muscles, and here this is the plant cell or stem or tissue. Now we can see here in details title stem cells are genetic or tissue engineering biotechnology in vitro, animal or human muscles or dental tissues productions, and class tissue productions for plant. Chapter 1, Introduction, Stem Cells and Cell or Tissue Regenerations. Before we discuss in short now, I can discuss a stem cell is a cell with the unique ability to develop into specialized cell types in the body. It can be animal cells, plant cells, or human cells. In multicellular organisms, stem cells are differentiated or partially differentiated cell that can differentiate into various types of cells. The process of reproducing or the state of being regenerated of a cell or tissue is called tissuous cell regenerations. The regrowth of a cell by an organs, tissue, cells, or any parts without seeds or that has been lost or destroyed is called regenerations. In biology, regenerations is the process of renewal, restorations, and regrowth that makes the genomes, cells, organisms, ecosystems resilient to natural functions or events that causes disturbance or damage. Here you can see agri-cats, they did these definitions. Tissue culture is a practice used to propagate plant, animal, human organs or tissue under sterile conditions, often to produce clones of a plant, animals, and human tissues. Different techniques in cell or tissue culture may offer certain advantage over traditional methods of propagations. Tissue culture relies on the fact that many cells, plant, animal, and human, have the ability to regenerate a whole organs of plant, animal, or human. This is called the totipotency. Single cells, plant cells without cell wall protoplast. Pieces of leaves or root cells, embryonic cells, muscle cells can be often be used to generate a new organs, tissue, or cultural media given the required nutrient and hormones. In the case of plant cells, animal cells, and human cells. Here we can see plant cell or tissue culture. Uh, Usually it is said plant cell or tissue culture if the sources can be used from the root cell or stem cell, then it is called the plant stem cell. And if it is used from the root, it's called plant root cell. Here we can see plant stem cells and tissue or cell regeneration class tissue regeneration here. Photo shows regenerated plants of tissues. Here we can see cell culture, yeast, and then animal cells and human cells. Image we can see here. In a laboratory, first started from plant, then animal, finally human cells. In a cell laboratory, I discussed before. First we start because it is easy to handle with plant cells. That's why when a new hormones or new chemicals comes to the market or invented, then scientists are trying to apply it to the plant first and then animal, then human. Plant cell and tissue culture, you can see here plant cells and tissue culture, plant cells, cell suspensions, and also tissue culture here, class formations, and from different plants, from pineapples, also other plants, banana plants, from tissues produce the whole plants and organs. So this is the from broccoli, shoot tissue culture by shoot stem cells, class formation and explants. You can see an image in my laboratory. We worked before. There's some image. We are working on the references. My name's also, my colleagues' name also here. We published many papers on these topics. Then chapter 2, we can in vitro meat productions. You can see here in vitro meat production introduction. In vitro meat is referred to the cultured meat, synthetic meat. It is an animal flesh product that has never been part of a living animal. This is the after slaughtering, the meat can be used in the laboratory. In the 21st century, several research projects have worked on in vitro meat in the laboratory. Baum, 2008, one scientist did this. The first in vitro meat burger created by a Dutch team was created as a demonstration for the press in London in August 2013, BBC 2016. There remains difference to overcome before in vitro meat becomes commercially available. Cultured meat is expensive, but it is expected that the cost would be reduced to compete that with that of conventionally obtained meat as technology improves. Jabs 2009. In vitro meat is also a cultural issue. This is the in vitro meats, in normal meats, and then this is of beef meat, and then we can see here in vitro we can produce the meat from this in the laboratory. So methods and methods. The process of developing in vitro meat involves taking muscle cells and applying a protein that promotes tissue growth. Once this process has been started, it would be theoretically possible to continue producing meat indefinitely without introducing new cell from a living organisms. So here we can see different scientists, they explained that report data. It has been claimed that conditions being dealt 2 months of in vitro meat production could deliver up to 50,000 tons of meat from 10 from muscle cells. We can use the pig, goat, any kinds of animals, camels. In vitro meat may be produced as strips of muscle fiber which grow through the fusions of precursor cells. So here same technique is the same. Almost in vitro meat may be produced in the laboratory. This type of meat can be cultured in a bioreactor or cell culture chamber or namely flow. Different types of chamber we can see here. Other cell types such as adipocytes would also need to be grown, and chemical messenger should provide clues to the growing tissue about the structure. So then we can see here different types of procedure of the laboratory. And this is the meat, beef meat can be grown in the laboratory. You can see here in the bioreactor also. So it can be grown in the bioreactor. Tissue engineering, biotechnology. This is for tissue engineering, biotechnology also. We can proliferation of muscle cells, proliferations of muscle cells in the laboratory. We can produce, though techniques is the same, the plant cell, animal cell, and human cells, but mode of action is different, or purpose is different. We can see there in details the commercial also now. Scientists are trying to produce commercially in different countries. So this is the character, the media and also other cultural media, and also this is the called cells sources and environment, and then bioreactor, nutrient and oxygen supply, the role of genetic modifications, techniques of genetic engineering such as insertions, deletions, silencing, activations or mutations of genes are not required to produce in vitro meat. Furthermore, in vitro meat is composed of a tissue or collection of tissues, not an organism. This is not a GMO, genetically modified one, but it is grown. Sometimes it can be GMO using different chemicals or hormones. Since in vitro meats are simply cell grown in a controlled artificial environment, some have commented that cultured meat more closely resembles hydroponic vegetables like rather than GMO vegetables. Dixon 2009. In vitro human meats also now produce in vitro human meats. This is the beef meats. This is the also human meat also for the organ transplantation or organ replacement. In some cases, for the human body, sometimes there is a lacking of the cells, the muscles. In that case, it can be used immediately. So this cells is just a human cells. It's important. So this is called the maternal cells. Different types of cells now different company is trying to produce in the laboratory. In the references we can see here, references. This is called chapter 3, also beef muscles generations by in vitro culture and burger productions. This is also new, burger productions in the laboratory. Meat made from small pieces of lab-grown animal muscle cells, or just a glimmers in some mad scientist eyes. That the in 2013 the dream of an artificial burger suddenly got interesting, and the Mark Post, the researcher, he invented this, and also his co-worker in the State University in the Netherlands, they produced this. Had created a burger made from real meat grown in the laboratory. They did this. Uh, they grow in the laboratory also. They produced the cells were placed onto the gel in a plastic dishes. This is the same procedure. Media, cups, serums, and media, which will eventually switch to a non-animal sources. So same media and same they used, and then finally they produced the meat in the laboratory. This is also once published in the New York Times. Published in New York Times newspaper, and then finally they succeed to produce meat and for burger. Lab grown meats. This is the lab grown meats. They produce the meat in the laboratory. You can see the picture. They also cultured meat for burger. They produce this in the laboratory from the meats sample. So here we can see, and they also lab grown meat, heated and edible meats. First meat production from lab grown meats. Dr. Mark Post, he invented this first, and then laboratory in the Maastricht University, Netherlands. He started this research and he succeeded. This is also beef muscle culture, beef muscle culture. Then culture beef and cow beef. This is cow beef and this is culture beef. Different types of beefs. Here we can see chapter 4, in vitro meat production from livestock animals. Also in the same procedure they used in the scientists are cooking up new ways of feeding the world's even growing hungers for meat that could save the environment and the lives of millions of animals. So in vitro meat productions, also Mark Post also he did this work and he produced, but it is still cost effective. That's why commercially can be produced. Most scientists are trying to produce commercially. Also in vitro meat muscle productions, in vitro meat in the laboratory. This is the references. Some references we can see here. In chapter 5, human muscle grown in laboratory, human muscles. It is very good attempt that lab grown muscles can be used commercially in the Europe. Laboratory first Duke researchers have grown skeletal muscles that construct and the response, just like native tissue to an external stimuli such as electrical pulses, biochemical signals, and the pharmaceuticals. The lab-grown tissue should soon allow researchers to test new drugs and study diseases in functioning human muscles outside of the human body. So this is the Nidan Barsik. He is associate professor. Nidan Barsik. Nidan Barsik, associate professor. Dr. Nidan Barsik, associate professor of biomedical engineering at Duke University. He did these researches and then he made his report. He published his report and he succeeded two lab-grown human muscles bundles test two lab-grown human muscle bundles. He also lab-grown human muscles in the laboratory. You can see here. This is also human muscles. So sometimes suddenly accident happens, occurred. In that case, muscles are needed for the patient, or sometimes muscle damaged in the body, skin, or any kind of muscles, skins, any kind of parts. In that case, if you grow these parts in the laboratory and it is ready to use, in that case, immediately we can use this organs or muscles from the laboratory for the patient. So that this also, he and his co-worker, they produced the many research. They did many research, and they produced the lab human muscles, and also other organs, human stem cells. So we can see here embryonic, then human embryonic stem cells. Embryonic stem cells are pluripotent stem cells derived from the inner cells mass of a blastocytes, an early stage pre-implantations embryo. This is from using the embryo, then can be used. It can be produced. Pluripotent also. This is another cells, embryonic stem cells of inner cells muscles are pluripotent. That is, they are able to differentiate to generate primitive ectoderm, which ultimately differentiate during gastrulations into all derivatives. So this is, we can see in the report, and they describe this, the report also here. I can see. Let's move to other topic, propagations, the same, and then apply. Embryonic stem cells are capable of propagating themselves indefinitely in an undifferentiated state and have the capacity, when provided with the appropriate signal, to differentiate. Presumably via the formation of precursor cells to almost all mature cells phenotypes. You see here application, potential clinical use. Usually the potential clinical use can be used clinically, and then repair of DNA damaged, differential somatic stem cells and ES cells, embryonic stem cells. Use different strategies for dealing with DNA damage. Sometimes when muscle damage, DNA also damaged from the portions. In that case, we call DNA damaged, so it can be repaired by replacing the muscles or the organs. So this is, we can see here, different scientists, they stated their reports about this embryonic stem cells. Different scientists, they report embryonic stem cell from other animals also, different types of animals. Other scientists, they produce this from Martins 1981. He produced also other new cells from their potential methods for new cell line derivations. So they mention the methodology. So almost the same methods, but sometimes it is little bit modification or some modifications. So then induce pluripotent stem cells contamination by reagents used in the cell culture. So that's why before use we have to this is called sterilizations. We have to make sterilization before use. The references we can see, there are some references on this chapter. Many scientists they did this work. And then stem cells and medical biotechnology. Here we can see stem cells and medical biotechnology. Stem cells refer to the cells we discussed before as the ability to divide continuously and differentiate develop into various kinds of cells. Rich 2004 he mentioned this. And then different types of cells: embryonic stem cells, fetal stem cells, umbilical stem cells, placental stem cells, adult stem cells, different types of cells. Yeah, they mentioned. And many scientists they mentioned their report, published report here, and their data. And there are also adult stem cells in humans. There's some bone marrows, adipose cells, liquid cells, blood cells. Some scientists use this type of cells for stem cells. So any kinds of parts of the plants or animals and human body can be used for cell culture. So here this is the image of the some cell culture lab grown. You can see here. This is also image of the muscle culture in bioreactor. Muscle culture in bioreactor. So references, there is some references here. You can see different types of reference. So stem cells from skeletal muscle cells and human muscle from skeletal other parts it can be used. We discussed before. So you can see here many scientists they explain their result, and others we can see methods for culturing mouse and human embryonic stem cells for animal and human. Many scientists they are using on animals, other scientists are using for humans, other scientists are using plant samples. So skeletal stem cells they made their report here. So we can see here different types of reports. If we study, then we can understand. So finally we can say here function of muscle repair, functions in muscle repairs. When muscles cells undergo injury, we send satellite cells are released from beneath the basement membrane. They become activated and re-enter the cell's cycles. Then these dividing cells are known as trans-amplifying pools before undergoing myogenic differentiation to form new post-mitotic myotubes. This is one kind of just how it works I mentioned. The mechanisms, now references here we can see, and some references. There is some for this chapter. Then let's move to other topic. Chapter 8: stem cells and influence on dental treatments. This is also a good topic. In dental treatments also there is stem cells and its influence on dental treatments. Stem cells are a key building blocks of human lives. Advances in stem cells research are bringing American medicine closer and closer to the day when the scientists told it will be used cell-driven therapies to attack diseases or repair damaged body parts. So this is the other types of stem cells. some scientists did this work. This is called the stem cells might help us when they repair damage to the teeth and gums. Teeth and gums also regrow, regrow, and they replace the jaws and the bones of the patients who have lost their jaws bones due to the cancer, traumatic injury, or developmental deformities. Deformities. So this is called the stem cells for dental treatments. Also important. They mentioned their data and they explained how they did this work. Dental stem cells for treatments and repairs. Some scientists. Rosnack et al. 2015. This is called the Virginia Commonwealth University. He did this research in this university, and others also. He did research School of the Engineering, School of Dentistry. Dr. Lau Lin, he did this research for dental stem cells for gums and the jaws. So he reported his results and he got positive results. He explained his recommendations. And University of Michigan also. Doctors at the University of Michigan School of Dentistry successfully generated patients jaw bones with the use of stem cells. This is jaw bones stem cells. This is also for dentals. Nowadays, new work on innovative. So still it is scientists are trying to make it to apply it in the clinic. University of California, Los Angeles also. They did some research on dental stem cells, gums and jaws, and they did genetic engineering technology, and they used the scaffold. Might be, and the same techniques they use stem cells could be activated in the patient's bodies. They might be paired with the engineered phosphate scaffold, essentially rigid structure, and bone can grow around. This is called the techniques in most of Southern California. They did also this work for dental treatments, tissue regeneration procedure. So this is the almost, you can see a bone and gums tissue should be fit snugly around the teeth like a third necks, pola around the necks. When anyone has periodontal disease, the supporting gums, tissues and bones is destroyed. Eventually, if too much bone is lost, the teeth may have to be extracted. The regenerative procedures are often recommended to gain some of the lost hard bones or soft gum tissues which have been lost. Here we can see they did this research work. So it is their report data they mentioned, and the scientists Jean and the teams 2003 and Rasnek 2015 they did this work. Also here we can see different types of the lost, and then they repaired this lost, and they repaired this by stem cells. So this is the results of the scientist. The scientist references, some references here you can see. So biography at a glance here. This is my essay next 25 Google Scholar, total citation 2495. This is the essay index 24 in the Scopus total journal and books 2016 216 I published books and journal papers. Then I said Web of Science papers 54 and conference semesters proceeding 54, PhD student supervision 6,
editorial board member, 7 journals, research project completed 19, external examiner for PhD thesis 5 in Saudi Arabia, Malaysia, and Bangladesh. So this is the almost my link, different types of link. So here we can see my contact also, email contact you can see here. And this is my published this book, Cell Biology, Structure, Function, Signaling, Cell Physiology, Regeneration and Inhibitions. With the colleagues, 3 colleagues, with my colleagues animal, he is working on animals, another working on humans, human cells, also principles of biotechnology. We published this. So we published cell and tissue culture, stem cells. Then we published plant biotechnology and genetic engineering. Thank you very much for watching, listening, and keep in touch. And also, I request you to be subscribed in my channel to get innovative informations. Thank you very much for watching.